Hello everyone. So this is biochemistry, Aiims capsule recording. Let's see the pattern of questions which are basically nowadays being asked in Aiims. This is one latest question from Aiims where they are talking about certain disorder and they're asking which may be the possible diagnosis. The presentation is frontal head enlargement, peculiar facies, thickening of cardiac valve leaflets, and hepatosplenomegaly. What is catchy point in this question is that cardiac valve is involved and organ involvement is there, hepatosplenomegaly is there. Child looks mentally retarded, frontal head enlargement is seen. So what may be the probable diagnosis? Answer for this question is going to be Hurler syndrome, which is one of the mucopolysaccharidosis. Hunter syndrome, you have no involvement of cardiac valves, you do not have involvement of cornea. So answer is Hurler syndrome, which is having more severe presentation compared to Hunter syndrome. When we talk about mucopolysaccharidosis, this is a very important table to be learned. In this, you see Hurler syndrome is having the presentation where opaque cornea, mental retardation, hearing loss, mitral valve involvement, aortic valve involvement resulting in regurgitation is all seen. Hunter syndrome, you don't have cardiac involvement. Tay-Sachs disease, you do not have hepatosplenomegaly, that I'll be coming to that slide little later. Fragile X syndrome also you do not have organomegaly. So the answer is Hurler syndrome in this case. Mucopolysaccharidosis is of so many types. Type 1 is the Hurler syndrome, which is characterized by deficiency of iduronidase. In severe deficiency, this is Hurler syndrome, and if the mild deficiency of iduronidase is there, you get to have this type 5 mucopolysaccharidosis, which is Shee's disease. When we talk about sphingolipidosis, we have so many types of sphingolipidosis. Tay-Sachs disease is one of them. You do not have organ involvement. Hepatosplenomegaly is not there in this particular disorder. Remember this table also, this is important. Tay-Sachs disease, Fabry's disease, metachromatic leukodystrophy, Krabbe's, Gaucher's, Niemann-Pick and Farber's disease are all different types of sphingolipidosis where sphingolipids are getting accumulated. You remember this feature of sphingolipidosis out of various sphingolipidosis which you learn. Hepatosplenomegaly is not seen in these two disorders, Tay-Sachs disease and Fabry's disease. This you remember. Cherry red spot is an important finding in certain sphingolipidosis. It is seen in Tay-Sachs disease, Niemann-Pick disease, metachromatic leukodystrophy, Sandorf's and Farber's disease. And where it is not seen, it is not seen in Fabry's and Gaucher's disease. So these are catchy points which you should remember. High chances of these questions coming in the exam is there. So cherry red spot is not seen in Gaucher's and Fabry's disease. Now these are core important points of these storage disorders. Niemann-Pick disease, you get zebra bodies, foam cells, macrophages. Gaucher's disease, you have crumpled tissue paper appearance. Tay-Sachs disease, you have onion layer skin, whole membrane with the lysosome. Krabbe's disease, you have multinucleated globoid cells. Metachromatic leukodystrophy, you have Hirsch-Pfeffer reaction. So these are important pathological finding, histological finding in various storage disorder. This is the slide which is discussing you various appearances. Gaucher's disease, crumbled tissue paper appearance, foam cell, bubbly appearance foam cell, Niemann-Pick disease, globoid cells in Krabbe's disease. Metachromatic leukodystrophy, the staining of brown color is seen. Zebra bodies in Niemann-Pick disease and onion layer appearance in Tay-Sachs disease. Try to remember these findings. So storage disorder is certainly important aspect of such exams. Aiims. So kindly revise all the tables of mucopolysaccharidosis and sphingolipidosis. Glycogen storage disorder is certainly important. That obviously by default you should remember, but what is more important that you remember, mucopolysaccharidosis and sphingolipidosis. Coming to various protein motifs. Protein motifs are the special conformations in the protein which are important role to play, which have important role to play as far as binding of these proteins to the DNA is concerned. We have zinc finger motif. Here the polypeptide is in finger shaped, it's looped like this, and polypeptide is looped like this. In the center you have zinc. So this is called zinc finger motif, where you can see this is structure. This is in the central area you find zinc, and the zinc is associated with the cysteine and histidine residues over here. This kind of appearance is seen in certain proteins, like for example transcription factor 3A, SP1, galactose 4 yeast protein, steroid H receptor superfamily. There are direct question to you that which all proteins have zinc finger motif, so try to remember these examples of proteins which are having zinc finger motif. Helix turn helix motif. HTF motif is helix turn helix motif. HTH motif. This kind of motif where you find alpha helix and turn so that the polypeptide is turning the direction of folding. So this kind of HTF motif is seen in lactose repressor molecule and homeodomain protein. Helix turn helix motif seen in lactose repressor and homeodomain proteins. The similar motif called helix loop helix motif. Helix loop, where one loop kind of thing is seen which is connecting two portions of helix. Helix loop helix is seen in MyoD protein, MYC protein and MAX protein. So try to remember the examples. When we talk about leucine zipper motif, this is another motif which is seen in proteins which are having tendency to bind with DNA. Leucine zipper motif, it's called leucine zipper motif because the appearance of the protein is having like zip, which is little unopened, and it's rich in leucine. It's called leucine zipper motif. And protein showing leucine zipper motif are phos protein, Zun protein and CREB. Cyclic MP response element binding protein. Cyclic MP response element binding protein. This is an important name they have asked twice. That leucine zipper motif is seen in CREB, phos protein and Zun protein. Try to revise all these things. Now this kind of question where they're talking about diabetes. A diabetic patient presenting with polyuria, blood glucose is 240 mg per deciliter. Which of these is likely to occur in this patient of diabetes? So in diabetes we know that there is lack of insulin. So what is seen in, that is what they are asking, in lack of insulin what is likely to occur? The answer is going to be increased conversion of fatty acid to acetyl-CoE by a process called fatty acid oxidation. Fatty acid oxidation. Fatty acid oxidation is the process which is taking place in lack of insulin. In diabetes you have lack of insulin, so there is fatty acid oxidation going on. So this is the answer in this question. Otherwise, if you talk about protein synthesis, if you talk about glycogenesis, whether in liver or muscle, both the places you need insulin. Glycogenesis demands insulin, protein synthesis also demands insulin, and cholesterol synthesis also demands insulin. So these pathways will be down regulated in diabetic patients. The protein synthesis, the glycogenesis and cholesterol synthesis will be down regulated, and what is seen to be there in diabetic patient will be increased conversion of fatty acid to acetyl-CoE. In nutshell, you should remember the important role of insulin in our biological system. Number one, it is glycogenic. Insulin is glycogenic. It favors the glycogenesis both in liver and muscle. Insulin increases glycolysis in liver. Insulin reduces glycogenolysis in liver. Insulin increases uptake of glucose by cells which are having GLUT4 containing cells. The GLUT4 containing cells are adipose, heart and skeletal muscle. These are cells which are having GLUT4 distribution, and GLUT4 is the only GLUT which is dependent on insulin. I'll be giving you a slide of GLUT distribution. So GLUT4 is found at adipose, heart and skeletal muscle, and the only GLUT which is insulin dependent is GLUT4. So these organs uptake the glucose in an insulin dependent manner. So what you have seen, insulin is glycogenic, it increases glycogenesis, decreases glycogenolysis, increases glycolysis, increases uptake of glucose by the cell. And when you talk about gluconeogenesis, insulin decreases gluconeogenesis. Insulin is anabolic, it increases protein synthesis. Insulin increases fatty acid synthesis. Insulin favors storage of triacylglycerol in adipose cell. So in lack of insulin, triacylglycerol is breaking to release the glucose to release the fatty acid in the blood. So it helps in storage of triacylglycerol in adipose cell. That is the reason in diabetic patients the fatty acid is not stored, rather it is more of the mobilized. So that results in increased fatty acid beta oxidation. So these are important role which the insulin is playing. Insulin also increases the cholesterol synthesis. So insulin is anabolic hormone. It results in synthesis of so many compounds. So this is a very nice question which is talking to you about the role of insulin. So which of the following is likely to occur in patient of diabetes? Because it's having lack of insulin, the fatty acid conversion to acetyl-CoA will be seen. Means fatty acid oxidation will be enhanced. Coming to next question, which is recent question of Aiims. Such type of questions are frequently asked. Cerebrohepatorenal syndrome is other name for Zellweger syndrome. This is a storage disorder which results in neurological dysfunction. In this, very long chain fatty acid is stored. Very long chain fatty acid is stored. Very long chain fatty acid uses peroxisomal enzyme for its initial breakdown. Whenever we are talking about very long chain fatty acid beta oxidation, initially that beta oxidation is taking place in peroxisome. There the very long chain fatty acid is trimmed in size, it's reduced in size. In Zellweger syndrome, what happens? The peroxisomes are there, but those peroxisomes are not having enzymes which are needed for VLCFA beta oxidation. Enzymes of peroxisomes are absent. The enzymes are formed, but they are not reaching the peroxisome. So peroxisomes are empty. So VLCFA, very long chain fatty acid, they do not undergo oxidation in peroxisome due to lack of enzyme, and they tend to accumulate. The predominant organs in Zellweger syndrome which are involved in this accumulation is brain, liver and kidney. Brain, liver and kidney, and even skeletal muscle is involved. That also is depositing lot of very long chain fatty acid. So which of the following substrate is accumulated? Very long chain fatty acid is accumulated. You should remember that Refsum's disease is another one problem where phytanic acid is accumulated. Phytanic acid is branch chain fatty acid. This is accumulated in branched chain fatty acid, which is accumulated in Refsum's disease. Zellweger syndrome, is neurological disorder where very long chain fatty acid is accumulated. Refsum's disease, you have branch chain fatty acid which is accumulated. This is also related with peroxisome dysfunction, where the peroxisome is the site where alpha oxidation is taking place. So Refsum's disease also, you have association of this peroxisome because alpha oxidation as such is defective in this Refsum's disease. Early death is seen in Refsum's disease, and patient is having retinitis pigmentosa, cerebellar ataxia, nystagmus, and very early death. So they are frequently asked. The Zellweger syndrome, the Refsum's disease are frequent question to you. You remember this table? This is an important one. Jamaican vomiting sickness is a disorder. It's a condition when the medium chain fatty acid dehydrogenase activity is stopped. Jamaican vomiting sickness, when medium chain fatty acid dehydrogenase activity is inhibited by a toxin called hypoglycin, which is there in unripe fruit of akki tree. Try to remember this line. So akki tree, unripe fruit, is having one toxin called hypoglycin. So if that is consumed, that will inhibit medium chain fatty acid dehydrogenase activity. This results in hypoglycemia, vomiting, to the extent that it results in electrolyte disbalance and ultimately death. So if a person is consuming unripe fruit of akki tree, he is having this kind of copious vomiting resulting in electrolyte disbalance and death and severe hypoglycemia. This is called Jamaican vomiting sickness. So they just ask you what is the toxin involved in Jamaican vomiting sickness. You remember the name, it is hypoglycin. Zellweger syndrome, though we say that the enzymes are not correctly placed in peroxisome, at times there is lack of peroxisome which results in accumulation of VLCFA. And Refsum's disease, I was talking to you, the defective enzyme which is not there, which does not oxidize branch chain fatty acid properly, is phytanic acid alpha oxidase or hydroxylase enzyme. So try to remember these kind of questions are very, very important in AIIMS. Now coming to this question, which is again a very, very important question. Which of these amino acid does not enter the TCA cycle by forming acetyl-CoA via pyruvate? And answer is going to be tyrosine. Tyrosine does not produce pyruvate, does not produce acetyl-CoA via pyruvate. Rather, tyrosine is converted to fumarate, and after making this fumarate, this tyrosine is entering TCA cycle. In this context, it is very important that you learn various amino acids as how they are producing the intermediate of TCA cycle. This diagram is very, very crucial, which is explaining you the various intermediate of TCA cycle coming from different amino acid. If you talk about succinyl-CoA, this is coming from Vim, valine, isoleucine and methionine. Valine, isoleucine and methionine, they produce your succinyl-CoA, and that succinyl-CoA enters the TCA cycle. If you talk about fumarate, the tyrosine and phenylalanine provide you fumarate of TCA cycle. Alpha ketoglutarate is coming from glutamate, and this glutamate comes from arginine, histidine, glutamine and proline. Oxaloacetate is coming via aspartate, and asparagine is an important source of aspartate. Now coming to the question in this discussion, when we talk about pyruvate, the source of pyruvate which is going to get converted to acetyl-CoA and then the TCA cycle, the source of pyruvate are alanine, cysteine, glycine, hydroxyproline, serine and threonine. Hydroxyproline, serine, threonine, alanine, cysteine, glycine. So they are going to provide pyruvate, which produces acetyl-CoA and used up in TCA cycle. There are certain amino acids like isoleucine, leucine and tryptophan which provide acetyl-CoA directly, which enters the TCA cycle. Then leucine, lysine, phenylalanine, tryptophan and tyrosine are the important amino acid which produces you acetyl-CoA, which produces acetyl-CoA and then enters the TCA cycle. So this is how somehow you try to learn this chart. It's a very, very important chart which is telling you that what all things can provide you pyruvate and then acetyl-CoA. So if you see this question, which of the following does not enter TCA cycle via formation of acetyl-CoA? The answer is tyrosine, because we have seen hydroxyproline, glycine, alanine, cysteine, serine, threonine, all of them they produce pyruvate and then acetyl-CoA. The reactions by which these intermediates are formed by amino acid degradation are called anaplerotic reactions. Anaplerotic reactions are filling up reactions, which are reactions which are needed to give the intermediates of TCA cycle. So anaplerotic reactions provide the TCA intermediates. Non-TCA intermediates are converted to TCA intermediate by anaplerotic reactions. We have cataplerotic reactions which drags out the intermediate of TCA cycle to other pathways. So cataplerotic where succinyl-CoA is taken up in heme synthesis, cataplerotic where citrate is going out in the fatty acid synthesis and cholesterol synthesis. Cataplerotic reactions are called emptying reactions. So you should learn all these things. Now coming to certain undergrads, practical. This is an important test called biuret test. This is the negative biuret test and this is the positive biuret test. All the proteins which are having at least 2 peptide bond, they give you positive biuret test. All the proteins having minimal 2 peptide bond, they give you this biuret complex which is having purple color. This control is taken which is having just the biuret reagent, no protein in this. It is to differentiate the color, positive and negative color. So biuret reagent is taken as a blank in another test tube and that is compared with the test tube. If that test tube is showing you purple color, which is much different from this light blue color, it shows you about positive biuret test. This test is called biuret test because 2 urea, when they condense with copper, they also give you this kind of colored complex. So copper is coordinated with 2 peptide bond to give you this colored complex, and similar colored complex is formed when 2 urea is complexing with copper. This is called biuret test. This biuret test is done to find out, to rule in or rule out the protein in the urine. In nephropathy, for any reason, you get to have loss of albumin, so that can be detected by biuret test. This test is heat coagulation test, where in test tube the urine is taken which is 3/4 filled and upper third of the urine is heated, and if that upper 1/3 shows the curdy appearance, it denotes you the presence of protein in the urine. This is heat denaturation test or heat coagulation test. On heating, the protein undergo denaturation, unfolding, and many unfolded protein, they clump together to form you coagulated protein. So this is a very simple test which can be done using a candle. Upper 1/3 of urine is heated and that precipitates the protein, and lower 2/3 is used as a control to see the cloudiness in the upper 1/3 on heating. So lower 2/3 is used as a control in this test. In this test, after heating, 2% acetic acid is added in the urine, drop by drop. This acetic acid is playing dual role in this heat coagulation test. If the haziness which is appearing on heating gets cleared off, if the haziness is cleared off, it denotes that haziness was because of impurities in the urine, sulfate and phosphate. They also tend to precipitate on heating. So if you add acetic acid drop by drop and haziness gets cleared, it means that there is no protein, rather it is the impurity which is getting precipitated. On addition of acetic acid, if the haziness improves or the cloudiness becomes more clear, it denotes the presence of protein, albumin, because on addition of acetic acid, the pH of the urine, which otherwise is 6.5, on addition of acetic acid, pH is reduced, it goes to 4.7 level. 6.5 pH is the normal pH of urine. On addition of acetic acid, it becomes 4.7. So this is the isoelectric pH of albumin. So at isoelectric pH of albumin, the albumin is easily precipitated. So on addition of acetic acid, if the haziness becomes all the more prominent, it denotes you the presence of protein, albumin, and if it is the haziness gets cleared off, it denotes you the impurities present in the urine. So this is how the heat coagulation test is a very, very easy test which can be done at primary health care level to find out the kidney status, whether the patient is having nephropathy or not. Now this next slide is talking to you one very important point, that is the distribution of GLUT. GLUT is glucose uptake and transporter protein. I was telling you type 4 is found in muscle, adipose and heart. Additionally, you should know the brain is having type 1 as well as type 3. RBC is having type 1. Liver beta cell is having type 2. None of them except 4 are insulin dependent. I insist again that even the liver GLUT, that is GLUT2, is not dependent on insulin for uptake of glucose. The entry of glucose in the liver cell is insulin free. It's a different thing that once taken up by hepatocyte, the metabolism of that glucose is totally dependent on insulin. I repeat that uptake of glucose from the blood by the liver is insulin free. It does not demand insulin, because GLUT2 does not demand insulin. But the metabolism of that glucose which is going to go to the hepatocyte certainly demands insulin. So in case of diabetic patient, what is happening, there is no insulin. The uptake is not affected initially, but once the glucose which is there in the hepatocyte is not able to get metabolized due to lack of insulin, further uptake of glucose is affected. And so that is the reason the liver stops consuming the glucose from the blood, and that results in high level of glucose in diabetic patient. So that you should know. And one more thing you should know, that GLUT2 is having important characteristic that it is bidirectional. It not only takes up the glucose from the blood to the hepatocyte, but also it pours the glucose to the blood from the hepatocyte. For example, during gluconeogenesis and during glycogenolysis, when the hepatocyte is having glucose there, that glucose is coming out to the blood via this GLUT2. So GLUT2 is bidirectional. So it is involved in uptake of glucose from the blood, and also it is involved in secretion of glucose to the blood at the time of need. GLUT6 is having no transporter role. It is said to be coming from pseudogene, where the protein is having no function at all. GLUT5 is involved in transportation of fructose, so it is a misnomer. GLUT5 is not a correct name. GLUT means glucose uptake and transporter protein. GLUT5 is not transporting any form of glucose at all, so it is a misnomer. It is transporting fructose from the GIT mucosa to the blood. GLUT7 is interesting one. This is found at endoplasmic reticular membrane, and it is the transporter of glucose 6-phosphate. This we need to have, this transportation of glucose 6-phosphate from cytosol to ER lumen is required during the process of glycogenolysis as well as gluconeogenesis. Because it's very important that glucose 6-phosphate, which is formed by glycogenolysis or by gluconeogenesis, should get converted to free glucose before it can come out by GLUT2. So the glucose 6-phosphate should be transported to endoplasmic reticulum lumen, because enzyme glucose 6-phosphatase is found there. I hope you understand. This is cytosol, this is endoplasmic reticulum. I am talking to you about this transporter which is called GLUT7. This is responsible for transportation of glucose 6-phosphate to ER lumen. Their enzyme glucose 6-phosphatase is found, which acts on this glucose 6-phosphate, converting it to free glucose, which is secreted out of the cell. GLUT7 is also called as glucose 6-phosphate translocase. Glucose 6-phosphate translocase. So you should learn this table, it's an important one. Coming to another one table which is important in Aiims exam, proteolytic enzyme and their mechanism, how they cut the bond. Proteolytic enzyme, they cut the peptide bond. We have trypsin, chymotrypsin, elastase, cyanogen bromide, lysyl endopeptidase, carboxypeptidase A, B, and pepsin. Carboxypeptidase, they cut the carboxyl end. They are exopeptidases. Try to remember this table, how they are cutting. Trypsin is endopeptidase, chymotrypsin is endopeptidase, but they cut selective peptide bonds. For example, trypsin cut those peptide bonds where carboxyl group is contributed by arginine and lysine. Chymotrypsin cut the peptide bond where the carboxyl group is contributed by aromatic amino acid or leucine, methionine, asparagine. So like this you should remember. What is very important in this is role of cyanogen bromide. Try to remember that carboxyl group, if donated by methionine, that kind of peptide bond is cut by cyanogen bromide. I again repeat, if the peptide bond mein kya hai? What peptide bond? You have one amino group, one carboxyl group. One amino acid is giving amino group, another amino acid is giving carboxyl group. So if the carboxyl group is contributed by methionine, all those bonds where carboxyl group is contributed by methionine will be cut by cyanogen bromide. Coming to microarray. Microarray is a technology where DNA chip or microchip is used. It's based on principle of hybridization. Hybridization means annealing of two complementary DNA sequences. In this one, chip is used which is called microchip or DNA chip, which is having lot of immobilized single-stranded DNA. If you see, this is the chip. Now you see the color coding, it is a developed microarray plate. If you see the raw microarray plate, you do not find any color. You just have wells which are having coating on internal wall with the single-stranded DNA sequences. So this microarray is actually used for genotyping, mutation analysis, and gene expression analysis. And in this, what is being used is complementary DNA, which is stacked with fluorescent material. So microarray is a technology which is based on hybridization principle. You just get one microarray chip and pour the complementary DNA mixture into all the wells. You allow it to hybridize. And after washing, you see some kind of fluorescence color coming out of various wells. Colors are decoded and you come to know that which gene is expressed to what extent in this particular cell. So this microarray is a very, very important technology where multiple gene expression analysis can be done at a time. So you should know that this is the technology where multiple gene analysis can be done at a time. All those genes which are coated on the internal wall of these wells are analyzed simultaneously for any mutation in them or for their expression extent. Northern blotting is a technology where gene expression analysis is done. But this technology is having one major limitation, that single gene expression analysis can be done at a time. Multiple gene expression analysis cannot be done simultaneously. So that problem is tackled by microarray technology, where this is the microchip where multiple wells are there. All wells are having coating of a particular gene on their internal wall, and the floppy provided with this microchip tells you which particular well is having what gene coating on the internal wall. So that helps telling you the expression of the gene in that particular cell in that duration. So these are certain important topics which I have covered in Aiims discussion, Aiims capsule, considering these as a potential areas where questions are frequently framed. You have to talk about mucopolysaccharidosis, sphingolipidosis, I stress. You learn all these storage disorder. Then core points like hepatosplenomegaly not seen in Tay-Sachs and Fabry's, cherry red spots seen in and not seen in Fabry's and Gaucher's, and then protein motifs. What you need to remember, that which all proteins you find zinc finger motif. Try to remember their names. And the importance of these motifs are very simple. The importance is that suppose if any new protein is discovered from the cell, and while analysis they found that this protein is having one of these motifs like helix turn helix motif, HLF motif, or zinc finger motif, or leucine zipper motif, they come to know that this protein which they have found is certainly having important role to play in genetics, because they have tendency to bind with the DNA. So I should again tell you that importance of this motif is that if this kind of motif is found in any protein, it signals that that protein is having DNA binding characteristic, right? So these are the proteins which are having zinc finger motif. These are proteins which are having HTF motif, HLF motif. Try to remember their names. Leucine zipper motifs. They are frequent MCQ. CREB was asked many number of times. Then diabetes, always discussion of diabetes is very, very important in biochemistry, because whole of the metabolism is altered due to lack of insulin. Insulin is a very powerful hormone which regulates so many important metabolic pathways in our system. So I have given you in a nutshell the important role of insulin in biochemistry, that how they are important for glycogenesis, preventing the glycogenolysis, protein synthesis, fatty acid synthesis, storage of TAG, and cholesterol synthesis. So if you know these points, you can sort out questions like this. Okay, now coming to next question which I discussed, it's very, very important. Nowadays they ask you very frequently, the Zellweger syndrome. So it's they may ask you which all organs are involved in accumulation. So name itself is telling you cerebrohepatorenal, but in addition you should remember even skeletal muscle is also involved in. And the main problem what lies in this Zellweger syndrome is that peroxisomal enzymes are absent. So they are certainly produced by the endoplasmic reticulum, but they are not targeted to the peroxisome. Just like in ICEL disease, I talked about Zellweger, that there is a lack of peroxisomal enzyme. When you talk about ICEL disease, you have lysosomal enzymes which are not reaching the lysosome. So lysosomes are the primary organelle which are affected, because lysosomes are not getting their respective enzymes. The enzymes are formed in the endoplasmic reticulum, but they are secreted outside the cell. So targeting defect is there in ICEL disease. Okay, so in lysosome materials accumulate, which gives you inclusion body appearance. So it's a lysosomal storage disorder. So similar fashion, the Zellweger syndrome is also peroxisomal related disorder, where peroxisomal enzymes are not reaching the peroxisomes. Then I gave you this table, which is important table in Aiims, and very frequently they ask you hypoglycin, Akitri, all these things you should remember. What manifestation patient will have? Severe hypoglycemia, severe vomiting, which results in electrolyte imbalance. Okay, so you should know all these things. Now, which of these amino acid does not enter the TCA cycle? This is again a very important area where we need to talk about various anaplerotic reactions and various amino acids which are giving you intermediates of TCA cycle. So I gave you this particular slide to remember. Then certain undergrad practicals like biuret test, heat coagulation test should be known to you. The role of acetic acid in heat coagulation test should be known. It's having dual role. It brings the pH down, which reaches 4.7 level, which is the isoelectric point for albumin, so that enhances the precipitation. Then we have glow distribution. So another important aspect of Aiims question that you should know. Then proteolytic enzyme, though it's a very difficult table to remember, but you know, I insist on cyanogen bromide, and of course trypsin is important. try to remember their mode of action, which peptide bond they target. Then microarray, I said multiple gene analysis can be done at the same time, and this is based on the principle of hybridization. So with this, I stop and wish you all the best. Thank you.